But you actually spend a lot of your time studying bacteria, which are larger than nanotechnology. But I think you work in an aspect of bacteria that actually does work at that level. Can you talk about what you work in and why it'd be interesting to have you on a nanotechnology show?
So my work is very interdisciplinary. We work in between nanotechnology and the bacterial world. And the thing about the bacterial communication with which I work mostly within is that it happens on a nanoscale. Many of these molecules that are secreted to communicate, that the cells use to communicate with each other, are produced in nano levels. So it's nanomolar concentrations, and it's so small concentrations that it's amazing that it makes such a huge effect on us as hosts.
How do bacteria actually communicate? How does that work? And then what are they saying?
What they do is that they send signaling molecules out in the extracellular space. So it's all about sending out signaling molecules, receiving them, and then sending the response back. And all of this, the signaling out and the response, all of these molecules are called quorum sensing molecules.
Quorum sensing. Okay, so what do we mean by quorum sensing by bacteria?
So quorum actually comes from this ancient history of, or also from the business world, that we have a minimum person number that can take a decision in a meeting, for instance. And this has been adapted in microbiology, and more specifically in what is called social microbiology, where bacteria are able to decide based on not how many they are, but indirectly by the density of the quorum sensing molecules, these signaling molecules that are secreted in their space.
How are you essentially using the tools of nanotechnology to spy on the bacteria in the human body?
So we place artificial receptors or nanostructures that look like receptors. And in this way, whenever we have a sample with some signaling molecules, we would be able to capture these molecules that the bacteria were sending to each other, but in this way we capture them first.
What do we actually know from bacteria communicating?
The first thing I learned is that they are extremely complex. So this whole process of communicating, I thought in the beginning that it would be easy, like, you know, just like our language. I say something, you respond with something, but with bacteria, it's really depending on the context. We need to first of all understand when do they secrete something and in what context they secrete it and what are these molecules and in what quantities. And when we have understood that, then we are able to kind of put it all together like a puzzle and say, okay, in the beginning of an infection, they secrete this, they do it in these amounts, they do it differently if they're exposed to antibiotics, for example, and how do they do it then when it's late in the infection. And in this way, when we put the puzzle together, this is the work we can use for our benefit as humans.
Can you share an example of how bacteria communicate?
So one example is that within this quorum sensing system is that they can produce molecules for the public good of their local society. And in this way, they're able to collaborate. They just produce signals that don't just benefit themselves, but everyone in the community. And what happens sometimes is some cells figure out that if they don't produce, then they can just scout on everyone else's production. And what happens is that sometimes many cells can do that. They cheat. And when many cells cheat, we call it a cheater invasion. And this is a very interesting phenomena because sometimes they end up like having a community with a lot of cheaters, and that just collapses the community. So just thinking about the fact that in many ways they operate like us humans. They need to take care of the common good of their community for them to survive as a whole.
So by listening to bacteria and understanding how to communicate and understanding their conversations, we can actually translate that into advances in medicine and help us treat bacteria or deal with these infections?
We have a long way ahead of us to be able to translate everything and understand everything. But there are also other ways we can use this. I mean, just because they are able to communicate and to send these signaling molecules to each other, imagine that we are able to make them unable to recognize these molecules. Like if they're not able to listen to each other, then how will they communicate and how will they coordinate their activities? So this is a way to find alternatives to antibiotics.
So you're part microbiologist, part molecular biologist, part nanotechnology expert, and part code cracker? Like you're actually cracking a language.
We hope so. And it's, of course, a collaborative work. So I work with a lot of people in different fields. I have a great collaboration with medical doctors, from where we get samples from real patients and we follow how is the infection progressing.
So you're working on a technology that could one day allow the everyday person insight into these bacterial communications.
So the big perspective here is to be able to translate every single secretion into this intracellular space and be able to translate it into actions. So if we somehow are able to use a smartphone or combine it with these nanosensors and detect on the spot, we will be able to, first of all, diagnose early, and we will also be able to figure out what treatment is necessary here and tailor this treatment to the specific patient.